Okay, hi everyone. So this is going to be the second video of cell respiration. So in the previous video we went over glycolysis, and in this video we will cover pyruvate oxidation. So this picture should look very familiar. It's from your textbook. It's another overview diagram. And glycolysis happened in the cytoplasm, remember? And pyruvate oxidation is going to happen in the matrix, which is this space in the mitochondrion, okay? So before I get into pyruvate oxidation, though, I do want to cover what happens with the electrons, the very high energy electrons from the two molecules of NADH, because that's really important as well. So you'll notice we have these white arrows going from 2 NADH across the membrane, and then jumping on either the NAD shuttle or the FAD shuttle, and then from there the electrons are then carried into the electron transport chains and ATP is produced, right? So NADH and FADH2, they're both considered electron shuttles or carriers that shuttle very high energy electrons to the electron transport chains, which is very important in ultimately producing ATP, okay? So the electrons from this NADH will move into the membrane, and I'm going to, let me preface this by saying I'm going to make it very simplistic layman's terms here. So the electrons will then move across the membrane, and they will hop on either the NAD shuttle or the FAD shuttle, and basically it depends on which one gets there faster. Now, depending on which shuttle these electrons jump onto, that will actually play a role in how much ATP is produced by the end of the respiration process. So if the electrons jump on the NAD shuttle, that produces about roughly 2.5 ATP per NAD molecule. So if we multiply that by 2, because we have 2 NAD molecules, then we would produce 5 ATP by the end of that pathway. And then if the electrons jumped on the FAD shuttle instead, that would produce about 1.5 ATPs, but yet we have 2 FAD molecules, right? So that would be 3 total. So in other words, depending on which shuttle these electrons jump on, whether it's NAD or FAD, that will determine how many ATPs are produced. And you can see that there's a 2 ATP difference, right? Because if it jumps on NAD, it's 5 ATP. If it jumps on FAD, it's 3. So that's a 2 ATP difference. And that's where this range of 26 to 28 comes into play. It's based off of what happens right here in the very beginning of the process. Okay? And remember, this 26 to 28 is a really arbitrary number because depending on which textbook or literature you read, you'll see different numbers being produced from oxidative phosphorylation. The specific number is not so important. What's really important is for you to understand that the majority of the ATP that's made in the respiration process comes from oxidative phosphorylation. Okay? Alright, so again, we are focusing on pyruvate oxidation, which is happening in the matrix of the mitochondrion. Okay? So from here, remember, this is, well, we're talking about oxygen being present, right? Because when we covered glycolysis, that whole process can happen without the presence of oxygen. But everything else, pyruvate oxidation, the citric acid cycle, and then oxidative phosphorylation, all those parts of respiration cannot happen without the presence of oxygen. So we are assuming, in this slide, we're assuming that we have oxygen present. And this is a eukaryotic cell, because in prokaryotes, all of this would just happen in the cytoplasm. So in pyruvate oxidation in a eukaryotic cell, we have the 2 molecules, remember there's 2 molecules of pyruvate that are being transported from the cytoplasm into the mitochondrion, across the intermitochondrial membrane, and then into the matrix for further oxidation. Okay, so here we have our 2 pyruvate molecules, and they will move across the membrane via the help of a transport protein, because pyruvate is a charged molecule, so it's going to have to be transported across the membrane actively. Okay. And then once it's transported across the membrane, it will be converted into this acetyl-CoA intermediate, or coenzyme A intermediate. okay? And that happens via a series of reactions involving a series of different enzymes, okay? And so all of those enzymes will come together to form what we call the pyruvate dehydrogenase complex, and it's a multi-enzyme complex. So pyruvate dehydrogenase complex is going to be a very important enzyme for you to remember. We are going to have a case study on it, and I will also talk to you a little bit more about that enzyme as well, and specifically when it's malfunctioning, what can happen, okay? So remember this enzyme. All right, so pyruvate dehydrogenase complex, that's what will help these three reactions happening. So what's happening is first we have the carboxyl group from the pyruvate will be oxidized and given off as CO2, and that's part of the waste product. And then the remaining 2-carbon intermediate will also be oxidized, and the electrons will then be transferred to NAD+, getting ahead of myself. And then that will be stored as energy in NADH, okay? And then from there, coenzyme A will attach itself to the 2-carbon intermediate, and then the acetyl-CoAs from here will then enter the citric acid cycle, okay? And so from there, we will talk about the citric acid cycle in the next video.